So what I want to do today is sort of tell you about a very exciting collaboration we've had with Life Technologies on utilizing sort of genomic approaches to look at stem cell pluripotency. And so what I actually want to try to do is sort of give you a bit more details on the background, the rationale, why we think this is actually working, and sort of show you a couple of very relevant examples to sort of put this into context. And so, sort of just briefly to set the stage, so what we want to accomplish is basically, like anybody else in the stem cell field, is we have pluripotent cells, the embryonic stem cells that have and should have the capability to differentiate into all three germ layers, which by definition is what makes them pluripotent cells. And so anytime you have a new embryonic stem cell line, you have to ask yourself the question, is my cell line actually indeed fulfilling this criteria of being pluripotent? And similarly, when you start with a somatic cell and reprogram that cell to derive iPS cell lines, for any one of those, you'll have to ask that same question. And of course, as you also know, cells in culture, because they should be self-renewing, can be propagated for a long time. You can actually ask that question many times over, because anytime you've propagated your cell line for many passages, you want to still confirm that your cell line still has maintained these properties. Moreover, we actually want to also understand not only can my cell line differentiate into these germ layers, but also how well can the cell line differentiate and make these alternative cell types. And so just as a sort of brief example, and most of you know that, but basically when we do these experiments, what we and many others are usually still doing these days is, and unfortunately you can't see a lot of the immunostainings on the screen, but you might be familiar with these pictures. So you basically, in order to test whether your cell lines are pluripotent, you first put them through an array of immunostainings for different pluripotency markers, surface markers such as the TRAS and SSA markers, or OCT4. On top of that, and again I apologize you can't see this, but basically on top of that you differentiate your cells into embryonic bodies. You might plate them as sort of monolayers, then differentiate them into specific cell types, and again immunostain them with another battery of different markers to just confirm your cell lines make all germ layers. Finally, sort of going from in vitro, you can also go and inject your cells into immunocompromised mice and generate teratomas to, again, test their three germ layer differentiation potential. Now, the issue with that is that, in particular in the human system, which we're talking about today, is that in contrast to the mouse, you're still not talking about extremely stringent assays. And so while in the mouse you can make a chimera or tetraploid embryo mutation embryos, in humans, again, it right there ends at the teratoma assay, which again, depending on whom you ask, and there was a very entertaining piece in Nature Medicine a while ago where you could find many interesting quotes like this one from Owen Witte on the controversial nature of that assay. That being said, the assay is useful, but it's not a very easy assay. It takes a lot of time, effort, and money. And so Peter Andrews actually put it very nicely, that what we actually want sort of in the field is something where you actually have a simple and robust assay that in a very quantitative way would allow you to compare different cell lines. And so this is what I'll talk about today. And so the idea is here obviously that these markers that we're actually measuring in these different assays, whether it's the surface markers or any of the pluripotency markers, obviously the expression of those is obviously tightly regulated and the result of well-defined gene expression programs. And so the idea is that if we can actually understand these gene expression signatures well enough, we can obviously make these correlative statements about when we see certain gene expression patterns in the undifferentiated or differentiated cell types, that we can actually now make predictions about what these cells can or cannot make. And so this, in fact, sort of going again from this experimental to a more genomic view, should again fulfill this criteria of being more quantitative and also therefore being more comparative between different groups. All right, and so sort of to sort of just give you a couple of examples for why we also think about this second point, that the actual ability and the efficiency to differentiate. So some of this, and in fact most of the interest in our lab for this particular question comes actually from older experiments from other groups, including this one from Doug Melton's group that was published a few years ago now. And so they made this very interesting observation when they differentiated different human embryonic stem cell lines into the endoderm lineage and then down to PDX1 positive cells. They saw very efficient differentiation of some lines, like in this case Hughes 8, but then other lines like Hughes 6 actually entirely failed to generate these sort of downstream differentiated cell types. And so we were very curious about this. This is sort of how we got actually into that particular genomic characterization, because we actually then started out using the Hughes 6 and Hughes 8 cell line, and we took a closer look at it, and by mapping different chromatin modifications as well as DNA methylation in these two cell lines, and also comparing to H9 cell lines, we immediately picked up hundreds of differences where, again, specific chromatin marks such as the K4 methylation, you can see the mark is entirely absent in Hughes 6 and it's present in Hughes 8 as well as in H9. And you've actually replaced this now by a stable repressive mark such as DNA methylation. So you can't see this, but this is a cluster of different zinc finger proteins. And again, we find hundreds of differences like that between these two cell lines. Now, we still can't link many of these directly to the phenotype, but at least we can sort of see very specific differences among them. And in our own experiments, again, when we differentiate different cell lines into endoderm and quantify it using CD184, which is a surface marker for the early endoderm, again, we see, you can probably not read these different cell lines here, but again, between the same cell lines, in many cases, we see consistent efficiencies, but actually quite different from other cell lines. And so, again, the goal is to try to get a bit more into why we actually observe these differences. And so, based on these sort of observations, we then sort of went back and said, okay, let's take a large panel of different human embryonic stem cell lines. And I'll talk about iPS cell lines in 1 second, but let's start with a huge panel of different human embryonic stem cell lines. And first understand, both on a gene expression level as well as an epigenetic level, what are the differences among these cell lines. And so, what you see here is, again, about 20 or so human embryonic stem cell lines plus some somatic fibroblasts as controls, global DNA methylation profiling, as well as gene expression profiling. Now, it's very hard to see on this. I mean, obviously, the pluripotent cell lines cluster both on DNA methylation and gene expression. They cluster together and away from the fibroblasts. But it's very hard to actually see at this resolution any meaningful differences. And so what I'll actually do in the next slide is just take these 15 genes, which sort of give you a spread from the left to right for low to high levels of DNA methylation, and then show you in a box plot the differences for these 15 genes across the 20 different cell lines. And so this is what's shown here. And so there's a couple of interesting things you can immediately see from this. And so you can see that there's some genes here on the left side, and so the legend is shown here, but you can see basically there's some genes in the genome where if you profile DNA methylation across all 20 cell lines, you actually see very little, almost no variation across these. So these genes are always unmethylated across all the different lines that we profile. On the other end of the spectrum, you see these other genes here where you see actually a wide range of DNA methylation values. And again, the same is true for gene expression. Wide range of DNA methylation values across these different cell lines. And so we can actually take advantage of this because based on this, because this is for the 15 genes, as I showed you before, we have this for the entire genome. And so now you can sort of define a range which you find sort of as an acceptable range, and then easily define outliers that are actually sitting outside of that range as sort of not fitting your standards for, in this case, DNA methylation, but again, same thing is true for gene expression. Now, interestingly, when we applied this, and apologies for this being a bit smaller, but now it's the same box plot, and now on the left side of the box, we're plotting the individual ES cell lines, and now on the right side, bringing iPS cell lines into this discussion, we're plotting different iPS cell lines. And so the most interesting thing you can immediately see from this is that genes in the genome that show very little variation in the ES cell lines, which are on the left, also show very little variation in the iPS cell lines. On the other hand, if you go further to the right, again, you can see now that lines that show wide variation in the ES cells also show a lot of variation in the iPS cell lines. And so what this immediately suggested to us that there's actually no fundamental differences between the ES and iPS cells, but rather we just observe a lot of natural variation that for whatever reason occurs in pluripotent cells themselves. And so that's an important point to understand, that we're not dealing with differences between these two types of cells, but rather something that's inherent to the pluripotent cell culture. Now, it also has obviously important implications, and I just want to show you two examples for the H1 cell line, because that's a line that's very frequently used in the field sort of as a reference for many experiments. And so the danger of using a single line as a reference set is obviously very easy to see here. So I'm just showing it for the 4 genes on the right here, which show a lot of variation. So you can see H1 sits at the bottom and at the low methylation end here for these two, but at the high methylation end for these two. And so to zoom in a little bit further, so let's say for CD14, again, H1 actually shows this very high level of DNA methylation. Now, if you compare this to a couple of iPS cell lines, you would clearly detect a significant difference between these two. That being said, if you look at more ES cell lines, you see that that's probably not a true difference between these cells, but rather something specific. But it's a meaningful difference because, and apologies, it's a bit small, but if you actually differentiate your ES cells into EBs and then further into sort of a macrophage-like cell type, we can actually see cell lines that have high levels of DNA methylation actually don't induce CD14 very well. So it has actually consequences for the induction of these genes. And another example, which is just shown here, just to make the opposite point, for the CAT gene, you see actually H1 is at the bottom end here, unmethylated, but there's lots of other ES and iPS cell lines that show high levels of DNA methylation here. And so when we now look at the correlation between DNA methylation and gene expression, for H1, you can see actually you can see the gene is actually being expressed and unmethylated versus another cell line, HUS1, which is actually now sitting at the top end of the spectrum. You can see even when you induce it in EBs, it stays methylated and not induced. And so we can very clearly see sort of differences between genes that become sort of apparently methylated in the cell lines and then no longer induced. Now, we can obviously, on a genome-wide scale, we can sort of, for DNA methylation, then bringing back the gene expression, we can obviously quantify this. But what we actually really wanted to do is, because that's sort of in the undifferentiated cells, and it's very nice that we can see these differences and we can even make conclusions in predicting forward whether or not these cell lines will differentiate into these particular cell types or induce at least a subset of the markers. But we wanted to do a little bit better, and so the idea was to actually see whether we can pick up gene expression signatures in just immediately differentiated cell lines, cells when they form initial EBs. And so the idea was, again, to try to find a quantitative signature in sort of this early EB differentiation. And so initially we had set up with a sort of panel of 500 plus genes. We basically profiled these, came up with signatures and scores. And so the interesting result was then when we took these scores of the ES cell lines or iPS cell lines and then correlated them with the actual direct differentiation, in this case taking the iPS cell lines and in collaboration with Kevin Egan, actually differentiating them all the way down to motor neurons, and then basically correlating our neural lineage differentiation score to the motor neuron efficiency, we saw a very striking correlation between these two, suggesting that we could pick up a gene expression signature that would allow us to basically predict and correlate the efficiency to make these motor neurons. And so that was also important because it was specific to the neural differentiation score, but not to the mesodermal or endodermal scores that we obtained. Okay, and so the idea is that we can use these gene expression signatures to basically predict and quantify the differentiation of these cell lines. Now, I do want to make one important point about that, just to sort of set the expectations right. And so that is, as you're all familiar with, obviously, you can reprogram cells with the overexpression of ectopic factors such as OCT4, SOX2, KLF4, and c-MYC to iPS cell lines. And moreover, you can reprogram pretty much any cell type into anything when you overexpress any other transcription factor. And so that just means that if you apply enough force, in this case transcription factors, or it might be other extrinsic signals, you can obviously make any cell type become anything. And so what we are measuring here is sort of not something that's not absolute, because you can obviously override this if you put enough pressure onto it. But rather, we're actually trying to have a very sensitive and quantitative way to indicate what are the actual propensities of these cell lines. And so that's just something to keep in mind when thinking about these assays. All right, and so obviously there was still some more work to do, and so basically a lot of the genes we initially picked didn't actually show a lot of interesting differences, and also the platform we had used wasn't actually very widely accessible. And so this is where we actually then teamed up with Life Technologies, and as a first pass, we actually sort of visited our gene selection and actually came up with a much nicer gene set of 48 genes that alone already had the same predictive power as the prior set that we were using. And then sort of went through, again, this sort of collaborative project with them, adapted it to the TaqMan platform. And again, there's a lot of sort of additional details on the downstream analysis that I won't be able to show you, that you should actually just check out on their booth. In addition to sort of this set, and again, sort of in collaboration with Life, we also added sort of a lot of additional insight that we had gained from a particular study that actually Casey Gifford was presenting on a poster yesterday. And so we could take advantage of a lot of high-resolution gene expression signatures that we had generated during the early specification into the 3 germ layers based on, again, signature profiling as well as RNA-seq, and sort of fill this up to a panel of 96 genes that we're now using in this platform. And so just to show you a couple of the trends, if you actually look at these genes and then group them into, let's say, pluripotency genes, endoderm, ectoderm, mesoderm genes, and look at a time course, this is showing, if you can't read it, it's showing you 0, 2 days, 5 days, and 12 days for these different groups of genes, in this case pluripotency and endoderm genes, and then for a couple of different cell lines. You can see very rapid and interesting dynamics and induction of these different gene sets for the different lineages, and again, you can see the same thing for mesoderm as well as ectoderm, showing that these genes are quite nicely induced during the differentiation. In fact, sort of you can zoom into this a bit more, color-coded in this previous way. And so again, this is sort of more addressing, coming back to the first question, where again, do we actually have 3 lineage specification in these? And you can see again, across this very large panel of different ES and also iPS lines, within 5 days, you can actually pick up very strong downregulation of the pluripotency genes and upregulation of these different, all 3 ectoderm, mesoderm, and endoderm specification. Now, obviously, on top of that, sort of knowing, so now you could very nicely see that you're inducing these different germ layers. You might induce them to different levels, and so again, for this, if you now, for example, take the day 5 as a reference time point, you can actually very nicely now correlate and compare your cell line to that new reference set that we have created. And again, it's still nice to see that if we pick up, sort of coming back full circle to the original example of hues 6 and 8 that I was mentioning, it's still nice to pick up that you see sort of this very biased and inefficient endoderm differentiation of hues 6, but much more efficient differentiation of hues 8 into endoderm that you again just pick up based on this gene expression signature and comparison. And so just to summarize that, so basically the main point was to try to figure out whether, again, we could use sort of these gene expression signatures, and I hopefully explained a couple of the details of why we think this really works, that you could now sort of reproducibly at different passages at any time for your newly derived or existing cell lines, basically create a measurement, and that's a measurement that if you would discuss it with anybody else or want to compare it or ship that cell line to someone else, they could run that and compare it to your results and see that they actually still work with the same line or at least a comparable cell line, which is sort of just important for actually comparing also the downstream results that you're obtaining. And so the one of the advantages now of this is obviously it's a more accessible platform than the previous one. This is sort of one thing we're very excited about, that you can actually pick up these signatures much earlier. So the prior set we were waiting all the way to day 16. So basically going down to day 5 is extremely useful. And of course having sort of more downstream and more adaptable computation and analysis tools built in is obviously a great advantage for sort of not computational savvy users that just want to run these analyses and get the results. And so again, you can sort of check out more details on that product at LifeTech's booth. And so with this, I just want to sort of thank a lot of different people. And so a lot of the basic work and sort of the groundwork on the ESL differentiation was actually done by Casey Gifford together with Michael Ziller. And then more of the specific collaboration between sort of life tech and developing this new panel was done by Alex Senkov, a computational biologist, and Veronica in the lab. And of course, I'd like to thank all the life tech team and my funding sources in general. And thank you for your attention.